So the title of my presentation is Silicon Transporters and Their Roles in Plants. So as all of you know very well, silicon has a number of beneficial effects on plant growth. It is especially important for sustainable and higher production of rice, because rice is a typical silicon accumulating species. Silicon is deposited at different organs. In case of rice, most silicon is deposited in the husk of rice grain, and this deposition is very important for higher production of rice. And this is a cucumber. As you can see here, is a white product. This actually is silica. So we call this a bloom. Yeah. Recently, the bloomless cucumber is more popular. This bloomless cucumber is produced by grafting the cucumber on a pumpkin root stock, which is defective in silicon uptake. As you know that the beer is rich in mineral silicon. So this silicon actually comes from the barley grain. Okay? And many foods contain a significant amount of silicon. So this means that silicon, silicic acid in soil, must be taken up by the root and then translocated from the root to the shoot and finally distributed to different organs. So all this process actually need different transporters. Okay? Different transporters. Without these transporters, silicon in soil cannot be transported from soil to different organs. So we are working on the silicon transporters. In 2006, we identified first silicon transporter, ELISA-1. Okay? And then in 2007, we identified the second silicon transporters, named ELISA-2. I just briefly touch ELISA-1 and ELISA-2. As you can see here, both ELISA-1 and ELISA-2 are localized at the exodermis and endodermis of rice roots. They show the polar localization. As you can see here, this is a double immunostaining. ELISA-1 is showing green color, and ELISA-2 is showing red color. You can see that the ELISA-1 is localized at the distal site, but ELISA-2 is localized at the proximal site. So they show the polar localization. And actually, ELISA-1 and ELISA-2 are completely different type of transporters. There's no any similarity between ELISA-1 and ELISA-2. ELISA-1 actually is an influx transporter. As you can see here, when you express the ELISA-1 in xenopus oocyte, you can see that it shows the influx transport, but the ELISA-2 does not show the influx transport activity. So this means that ELISA-2 is not an influx silicon transport. We found that ELISA-2 actually is an efflux silicon transport. As you can see here, when you express the ELISA-2 in the xenopus oocyte, it shows the higher silicon efflux transporters. So if you knock out either ELISA-1 or ELISA-2, the growth of rice was seriously inhibited. Actually, the yield, you got a great loss of yield. Actually, the yield of ELISA-1 or ELISA-2 mutant is only 10% of the wild type. So from this experiment, you can feel that how silicon is important for rice production. Because without the silicon accumulation, you can only get the 10% of the yield of the wild type. So this is the latest version for phylogenetic tree of ELISA-1, and this actually was prepared by my colleague, Dr. Naoki Yamaji. As you can see here, that in the higher plants, there are two, the ELISA-1 homologue can divide it into two different groups. One belongs to the monocots, and the other belongs to the dicots. And in the monocots, silicon transport 87 homolog has been identified from the maize, barley, and wheat. In the dicots, silicon transport 87 homolog has been identified from the pumpkin and soybean. Recently, a different group, ELISA1 influx transport, has been identified from the horsetail. So Richard always said that the horsetail is not a useful plant, but I want to tell him that this is very useful in Japan. It is used for the decoration, so it has a healing effect. So I want to tell Richard, don't feel so sad, okay? Because it's very useful in Japan. So as you know that ELISA1 actually belongs to the Aquaporin family. So Aquaporin family has a very big, family, and in plant it has 4 subfamilies. The silicon transporter belongs to NIP subfamily. This NIP subfamily is very specific to plant. It's not present in the animals and the mammalian. So as you know, in 2003, Peter Agri from the United States, he received the Nobel Prize because of the discovery of water channel, okay, aquaporin. So I also I'm dreaming, maybe one day in 2000 XX, the Nobel Prize goes to Jian Feng Ma for the discovery of silicon transporters. Compared to ELISA1, only limited numbers of ELISA2 transport homologs has been functionally characterized. As shown here, only silicon efflux transport from maize, barley has been characterized. In dicots, only ELISA2 from the pumpkin has been characterized. Most other ELISA2 homologs has not been functionally characterized. So these transporters actually are involved in the uptake, translocation, and the distribution. Because I talked about the silicon transport involved in the uptake at the last silicon meetings. So today I will focus on the silicon transporters involved in the distribution. But just to give you 2 updates about the silicon transporter in uptake. Recently, so far we use the cultivated rice, okay, for silicon transport research. So we want to know how about the silicon transport in wild rice, because wild rice, we don't know, they have the same system, efficient system for silicon uptake as a cultivated rice. So we cloned the silicon transporters from the wild rice with different genome, and just to give you one result, show you one result. This shows the silicon transporters in wild rice. They show you the cellular localization of ELISA1 and ELISA2. And so as you can see here, similar to the cultivated rice, in all wild rice, they have similar cellular localization of both ELISA1 and ELISA2. This means that this system has been conserved from the wild rice. So another update is we try to understand why rice has such efficient uptake system. So we are doing mathematical modeling. As a result, we found that the localization of ELISA1 and ELISA2 at both exodermis and endodermis are very important. And we also found that actually the Caspanian band strips is also very important for the efficient silicon uptake in rice. If you want to know I found that this is not the version I used for the presentation. Some piece is lost. Yeah, so we just submitted this work to plant and cell physiology, and actually this work is done by my colleague Gen Sakurai. Okay. So today I will focus on silicon transport involved in the distribution. So this is just to show you the flag leaf and the panicles of rice. As you can see here, compared to the flag leaf, the panicle has a much smaller surface area. So if the distribution of silicon follow the transpiration, more silicon will be distributed to the flag leaf, and the less silicon will go to the panicles. But if you analyze the silicon concentration in different organs, you will find that actually more silicon in the husk of the grain. So this means that the silicon distribution does not follow the transpiration. Okay? So previous physiological study has shown that the distribution is controlled at the node. Okay? So I hope you understand what I'm saying, because when I talk about the node, maybe my pronunciation is not so good. People always misunderstand me. Are you going to talk the noodle? Or I said no. They said, are you going to talk about the nude? So unfortunately, I'm going to talk about the node, okay? N-O-D-E. So node actually is a characteristic structure of graminaceous plants such as barley, rice, and wheat. In modern cultivated rice, there are 13 to 18 nodes, but most of them are sticked at the basal region. So only the up 4 to 5 nodes, internodes, are elongated at the reproductive stage. And the remaining nodes are sticked at the basal region. So each node actually has a similar structure, including one leaf or leaf tiller bud and crown rods. Initiate at the node and connected to the developing tissues such as young leaf and panicles. So diffuse vascular bundle initiate at the node and connected to the developing tissues such as young leaf and panicles. So minerals include silicon must be transferred from enlarged vascular bundle to diffuse vascular bundle to provide silicon to the young tissue and the panicles. Okay? So this process is called intervascular transfer. So plant has developed an efficient system for intervascular transfer. One is to increase the vessel area, and another one is to increase the uptake surface area. So this is a cross section of node. As you can see here, this is a xylem vessel. If you compare it to the up and the lower tissue, you can see that the vessel area is much bigger, larger than up and lower tissues. So this enlargement actually reduces the speed of mass flow and make nutrient, including silicon, stay longer at the node. So this is one strategy. Okay? So another strategy is there's a specific cell called Xylem transfer cell. Okay? As you can see here, this cell has a folded plasma membrane because of ingrowth of cell wall. So this folded plasma membrane increase the uptake surface, so make uptake efficient. So to understand what kind of silicon transporters is involved in the preferential distribution to the panicles. So we investigated the expression of many silicon transports in the node. So this is the data. As you can see here, at the reproductive stage, ELISA 2, ELISA 3, and ELISA 6, they show a very high expression level at the node 1. Okay? Node 1, as I told you, connected to the panicle and the flag leaf. So it is responsible for the silicon distribution. So then we characterized each gene and investigate what kind of role they are playing in the silicon distribution. So what is ELISA 6? So ELISA 6 actually is a close homologue of ELISA 1. As I mentioned, ELISA 1 is involved in the silicon uptake, the first silicon transport we identified. So we found that ELISA 6 actually also has an influx transport activity. So ELISA 6 is also a silicon influx transport. And where it is localized, so we did an immunostaining of ELISA 6. As you can see here, ELISA 6 actually is localized at the Xylem transfer cell. So this is ELISA 6, and freeze again. Yeah. If you look at this picture, you can see that the ELISA 6 show a polar localization. It facing to the Xylem vessel. So it is responsible for Xylem unloading of salicylic acid. So what happened if we knock out ELISA 6? So you can see that if we knock out ELISA 6, you got increased silicon accumulation in the flag leaf, but you got the decreased silicon accumulation in the peduncle, latches, and husk. So this means that silicon is involved in the distribution of silicon to the panicle or to the flag leaf. So this is actually a simple experiment. When you cut the panicles of wild type and the ELISA 6 mutant and leave on the table for just 3 hours, you can see the difference in the phenotype, because this is actually caused by the water loss. So you can see from this picture that the whole silicon is important for protecting water loss from panicle. Okay. We did another very simple experiment to show the role of ELISA 6 in the silicon distribution. We grew both wild type and the ELISA 6 mutant hydroponically in the absence of silicon till heading time, and then we just give 2 days. We just give silicon for 2 days, and then see. Then look at the silicon distribution at the different tissues. As you can see here, in the wild type, about 14% of silicon, of total silicon, was distributed to the panicles. By contrast, only 1.4% of silicon was distributed to the panicle. If you look at the leaf blade of flag leaf, you can see that the more silicon was found in the flag leaf in the ELISA 6 mutant, but the less silicon was found in the flag leaf of wild type. So this means that the knockout of ELISA 6 altered the distribution of silicon. Okay? So from this result, we can conclude that ELISA 6 localized at the Xylem transfer cell play a very important role in the intervascular transfer from enlarged vascular bundle to the diffuse vascular bundle. Especially, it plays a very important role in unloading salicylic acid from the enlarged vascular bundle. And then we also investigated other 2 genes, ELISA 2 and ELISA 3. Okay? ELISA 2, as I mentioned earlier, is an efflux, silicon efflux transport. It is also involved in of course, it is involved in the silicon uptake. Okay? And then we expressed both ELISA2, ELISA3 in the xenopus oocyte and look at the efflux transport activity. As you can see here, similar to ELISA2, ELISA3 also show the silicon efflux transport activity. So this means that both ELISA2 and ELISA3 are efflux transporter of salicylic acid. Okay? And then we look at the localization of ELISA2. And this picture shows the localization of ELISA2 at the node 1. The green color shows the ELISA6, and the pink color shows the ELISA2. As you can see here, ELISA2 is localized at the cell just next to the ELISA6. And if you look at it very carefully, you can also see that the ELISA2 show a polar localization. Okay? It is localized at the proximal site. And then we look at the localization of ELISA3 by using immunostaining. As you can see here, the green color is ELISA6, and this is ELISA2. The pink color is ELISA3. You can see that ELISA3 is localized between ELISA2 and the diffuse vascular bundle. Okay? So, what happened if you knock out ELISA2 and ELISA3? So we did a very simple experiment. Because ELISA2 is also involved in silicon uptake, so to exclude the lure of ELISA2 in the silicon uptake, we just cut the panicle from the second internode and then feed silicon from the internode, and then look at the distribution to the flag leaf and the panicles. As you can see in these figures, knock out ELISA6, ELISA2, or ELISA3 resulted in more silicon distribution to the flag leaf, but less silicon distribution to the panicles. So this result very clearly shows that this three transport, ELISA6, ELISA3, and ELISA2, play a very important role in the intervascular transfer of silicon. This is just a summary of ELISA6, ELISA2, and ELISA3 in the node 1. As you can see here, they are localized at the different cell layer. So ELISA6 function as an influx transporter, ELISA2 and ELISA3 function as an efflux transporter. So they are responsible for the intervascular transfer of silicon from the enlarged vascular bundle to the diffuse vascular bundle. If you knock either of them, they will change the distribution, and more silicon will go to the flag leaf, less silicon go to the panicles. So all these three transports are very important for the preferential distribution of silicon to the panicles. So as I mentioned that the node is a characteristic structure of gramineous plants. So far I just show you some data in rice. We also did some work in barley. Actually, barley also has a similar system as rice. So just to show you one slide, we did in barley node, as you can see here. This is ELISA6, actually from barley. It is also localized at the transfer cells. And this green one is ELISA6, the red one is ELISA2. You can see they are localized at the different cell, the next cell layers. So I think they are also play a very important role in the intervascular transfer of silicon at the node 1. Okay? Because of limited time, I'm not able to tell you all stories, but fortunately, we recently just published a review paper on the node in Trends in Plant Science, and we also have actually a video abstract. So if you are interested in node, just click YouTube and put Ma node, you can find this pictures. You can find this articles, okay? So acknowledgement. So this is our current lab numbers. I'm sorry. Acknowledgement. So this is the current lab members, and we are actually doing different mineral elements. We are working on aluminum, we are working on silicon, we are also working on cadmium, arsenic, zinc, manganese. So the work I introduced today actually was mainly done by my colleague Naoki Yamaji and by myself. You may not believe I'm still doing an experiment. Okay? And our funding is from the Ministry of Agriculture, blah, 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 and the Ministry of Education and Science, blah, 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 blah. So thank you for your attention.